This is a cancer cell. It's basically a vampire. Like regular cells, it grows and uses resources, but unlike regular cells, it's immortal and can grow literally forever. Most cancers get this immortality using a protein called telomerase. This little guy has been the favorite child of longevity gurus forever because of its ability to lengthen cells' telomeres and help them live longer. Most healthy cells don't express the protein part of telomerase, a protein called hTERT. However, 85 to 95% of cancers do. What if we could build a gene that detects this hTERT and kills the cancer from the inside out? That's what I did. Or at least, I designed a system that we could use to detect telomerase. In this video, I'm going to tell you about the science this system is based on, how I did it, and how I used Cadbury to design an algorithm that generates the sensor for me. This is an internal ribosome entry site, or IRES. It's a folded tangle of RNA that grabs the ribosome in the cell and forces it to start making the protein. This IRES is from hepatitis C, so it's called the HCV IRES. The important thing to note is that it's made of RNA, not proteins. This means that RNA can interfere with it too. Like DNA, RNA is made up of complementary bases. A bonds to U and C bonds to G. If another strand binded with the IRES, it could distort its shape in such a way that it couldn't do its job and bind to the ribosome. If RNA can interfere with the IRES, what if we could redesign the IRES to use this as a sensor? Well, that's exactly what my system does. I'm calling it TRIES, for Translation Regulation Using IRES Sensors. By the way, translation is a fancy biology term for when you turn RNA into a protein. And we regulate that using an IRES. We start by adding an attenuator after the 3' end of the IRES. The 3' end is the bit highlighted in red. The attenuator is made up of 2 bits that work together to regulate the IRES. First, there's the antisense region. This is a bit of RNA that's designed to be complementary to whatever target you're trying to detect. This means that if the target's present, it will bind to the target, changing its shape. Next, there's the regulator region. This is a bit that's designed to stick to the IRES and distort its shape. Together, these 2 parts can act like a sensor. Because double-stranded RNA is very stiff, if the target binds to the antisense region, it forces the attenuator region, the binding region that's trying to bind to the IRES, away from the IRES, and stops it from binding. Finally, to avoid having a bunch of random bits of protein at the start of your protein, I've added a 2A peptide, which forces the ribosome to skip a bond, splitting the protein into 2 different proteins. Okay, so it isn't actually that simple. When you're picking a region to target, you want to make sure that the attenuator doesn't interfere with the IRES on its own, because that's not the point. You also want to make sure that the active configuration, which is when it's bonded to the target and working, is much more stable than the inactive configuration. Otherwise, it'll kind of just float around between the 2 states. On top of all that, you need to make sure that the target is actually available to bind to the attenuator. It isn't going to be able to bind if it's already part of some much bigger structure in the RNA, and most mRNAs have a structure. Our algorithm needs to be able to consider all of these variables in a relatively quick amount of time. Because of this, I decided to go with a genetic algorithm. First, the algorithm uses an RNA library to fold the RNA target to pick out regions that it's able to target because they're free. Next, the algorithm generates a starting population of sensors. In each generation, it evaluates these sensors in parallel and uses the best ones to create sensors for the next generation. Finally, the algorithm mutates each of these sensors and adds some random ones each generation to introduce variation. The algorithm is fairly slow, mostly because of the way it evaluates sensors. It has to fold each sensor twice, and since they're about 350 to 400 base pairs long, that takes a while. I was able to run 100 generations of 100 sensors, so 10,000 sensors, on an 8-core Ryzen 7 laptop in about an hour and a half. As of the making of this video, the algorithm is live on the developer branch of Cadbury on GitHub. Be warned, it's the developer branch, so it might be unstable. Maybe I accidentally flipped all the RNAs in the wrong direction. That has happened before. It's released under the GPL v3 license, so feel free to do pretty much whatever you want with it under those conditions. Okay, so now what? We've got this sensor that works in theory, but where do we go from there? First things first, we need to test it and prove it isn't just some random idea I came up with. Luckily, the HCV IRES works in yeast, so it makes it a lot easier to test because we wouldn't have to use human cells. I outlined how I plan to test it in an article that I wrote and linked in the description. So, if I want to test it, I need a lab. Basically, I have two options here. I can either try and get into a legit lab, or I can build my own. And I might also be able to interface with some local DIY biology enthusiasts. COVID protocols are making it really hard to get into a real university lab right now, so I may have to go the second route. If I end up building a lab, stay tuned. I will definitely make a video about it. With that said, subscribe, keep informed about when I post a follow-up, because I will test this somehow. And with that said, thank you for watching. Have a nice day.